Okay, so we are recording and then we'll start with uh, the session. So, welcome everyone to the World Oceans Day webinar hosted by Ocean Society of India Pune chapter. We have with us Dr. Pamela Muller, and we are incredibly honored to have her speak here today. The World Ocean Day is celebrated every year on 8th of June, and throughout the year, the UN works with catalyzes on collective action for a healthy and stable climate. And this year's theme is essentially to work towards protecting our marine environment. So the action theme is strong marine protected areas for our blue planet, and it is supposed to guide global action over the coming years. It brings together a momentum of recent multi-year action themes like catalyzing the ocean climate action and protecting at least 30% of the lands and waters and the ocean by 2030. So to begin, I invite Dr. Supriya Chakravarti to please give an introduction about Ocean Society of India.
Nature. Good morning, everybody. So I like to
Mhm.
give you a brief introduction about the Ocean Society of India Pune chapter and its activities. The Ocean Society of India is a professional society that brings together people who are interested in or working on various aspects of ocean sciences, engineering, technology, and related fields. The society has live members from different parts of the country who are organized into local chapters in Kochi, Kharagpur, Chennai, Pune, Goa, Delhi, Visakhapatnam, and Ahmedabad. These chapters conduct regular activities such as seminars, workshops, lectures, and field visits to foster interaction and collaboration among the members. The main objective of OSI is to provide a platform for exchanging the first scientific and technological developments in the ocean domain and to facilitate networking and partnership among the stakeholders. The society aims to inspire and motivate working professionals, researchers, and students to pursue excellence in their respective fields and to contribute to the advancement of ocean knowledge and innovation. The society also strives to empower the community to protect and manage the oceans and their resources in a sustainable manner. The society organizes various educational programs, awareness campaigns, ocean conservation initiatives, success stories, and good practices to disseminate information and to raise public awareness about the importance and benefits of the oceans. The OSI is registered under the Societies Regulation Act 1860 and has its registered office at the Center for Marine Living Resources and Ecology at Kochi. The society is governed by an executive committee elected by the general body of members. The society conducts national and international conferences, symposia, and exhibitions on relevant topics in ocean sciences, engineering, technology, and allied fields. The upcoming biennial conference, OSICON 2027, will be held from January 21-23 at IIT Pune. Today, the Ocean Society of India is celebrating the World Ocean Day 2026 in a befitting way with several outreach programs. One of the programs is beach cleaning at selected locations in the Indian coastline. The OSI Pune chapter is an integral component of the OSI. The members of the Pune chapter are actively engaged in various outreach programs to popularize the awareness on ocean. It regularly arranges webinars and lectures by eminent scientists, occasionally in association with other professional bodies such as the IMS Pune chapter. One of the flagship initiatives of the OSI Pune chapter is its outreach program, the publication of Ocean Digest, the quarterly newsletter of the Ocean Society of India. This newsletter features popular articles on ocean science, engineering, and allied fields, along with updates on the society's activities, all aimed at fostering greater ocean awareness. The OSI members, especially the scientists and the students from IIT and SPPU, regularly contribute in this newsletter. The OSI Pune chapter members actively contributed to various initiatives of the parent body, including serving as election officer for the GC committee elections spanning the successive terms of 2023, 2025, and 2025-2027. Additionally, they played a key role in moderating the PG Dissertation Award for 2024 and facilitating the commissioning of the Professor R. Ramesh Endowment Award, which recognizes excellence in the field of ocean and climate science. The OSI Pune chapter welcomes anyone who is interested in learning more about the ocean and its resources. Thank you.
Thank you, sir.
Thank you so much, sir. So now I'll invite Dr. Rajni Panchang to introduce her host, Dr. Pamela. Please go ahead.
It is indeed a proud privilege to introduce world-renowned biological oceanographer, Dr. Pamela Hallock. Dr. Pamela Hallock Muller is better known in the Indian micropaleontology world as, as Dr. Pamela Hallock. She holds a bachelor's in zoology from the University of Montana, acquired in 1969, and a PhD in oceanography from the University of Hawaii in 1977. Her first academic position was as the assistant professor in earth sciences at the University of Texas of the Permian Basin. She joined as a faculty at the University of South Florida in 1983, completing almost 50 years, more than 50 years in this department. As an associate professor in marine science, she was promoted to professor in 1988. By studying larger foraminifera in the geological record in the modern carbonate systems, Professor Hallock and her graduate students gained insight not only into the environments of the past and present, but also the effects of human activities on the future of Earth's ecosystems. Her work has implications for reef and shelf ecology, environmental management, evolution, paleoceanography, paleoecology, and sedimentology. She has co-authored over 200 scholarly papers, has served as thesis or dissertation advisor to more than 75 students, and is a committee member for more than 60 others, and has hosted visiting scholars from around the world. We were just talking that in her house, she has hosted scholars from over 50 countries, of almost more than 50 scholars belonging to 22 different countries, which we this time actually listed out. She received the 1999 Outstanding Educator Award from the Association of Women Geoscientists, the 2012 Alfred Sloan Foundation Minority PhD Program Mentor of the Year Award, uh, and what people know about is her research has been honored by earning the 2015 Joseph Cushman Award for Excellence in Foraminiferal Research and the Raymond Moore Medal for Excellence in Paleontological Research by the Society of Sedimentary Geology. Her paper in 1986, Hallock and Schlanger, titled Nutrient Excess and the Demise of Coral Reefs and Carbonate Platforms, was named as one of the landmark papers in carbonate sedimentology and stratigraphy by the American Association of Petroleum Geologists 100th Anniversary Committee in 2017. She is the Fellow of the Paleontological Society and the Geological Society of India, and a sustaining member of the Association of Women Geoscientists, the Association of Women in Science, and the Society of Sedimentary Geology. She currently serves as a distinguished professor at the University of South Florida. One of her greatest contributions, which people know her for, has at least in India and Indian geological fraternity or oceanographic fraternity, has been her development of the Foram Index for monitoring coral reefs. She has been monitoring the Florida Reef Tract, the coral reefs of the Florida Reef Tract since the 1980s. She is 78. On 2nd of June she just turned 78, and she still scuba dives, and her experience of the coral reefs is what she sees with her own eyes and touches with her own hands. And her lab is called the Reef Indicator Lab, which many of us think she only works on foraminifera, but not just live and larger foraminifera, but she has several students who have worked on different components and different organisms that make up the coral reef ecosystem. With this little, and it is a proud privilege, and it is because of her that she hosted me that I think I was able to develop this program and also back the Fulbright Award that I have right now. Thank you so much, ma'am, and it's a privilege to have you here.
Well, thank you for that introduction. That was,
Yes, Aditi.
I think I gave you too much information.
But it is so great to know. Also, belated birthday wishes. And I was actually talking to Rajni that the last year and this year we both had women oceanographers speaking at our webinars, and it is
It just
an
happened, not
immense
planned.
honor. Yeah, that
Happened, not planned.
because we need inspirations, and all women scientists working in the field are a huge inspiration for women all over the world to get into this field. So thank you so much for agreeing. So without any further delay, we will start the presentation. So I'm just sharing my screen. I hope everyone is able to see it. Just give me 1 second.
Pam, you can just keep saying next and we'll change the slide.
Yes. Yeah. Can someone confirm if you are able to see the
Yes.
full screen?
Yeah, now it's
All
on
right.
full
Yes,
screen.
we are able
All
to
right.
see.
Yes, we
Great.
can
Yes.
see.
Thank you so much. So, Pamela, you can start. And to the audience members, may I please request that you check that you are on mute so that Dr. Pamela is not interrupted. And we will have a question answer session at the end of the talk. Thank you so much. Yes, Dr. Pamela, you can start.
Well, thank you for that lovely introduction, and thank you for the privilege of speaking with you all today. Um, what I'm going to talk about is a bit about my career, well, quite a bit about my career, I suppose, and essentially conundrums or paradoxes that uh, we as scientists sometimes or oftentimes encounter, and oftentimes, well, I'll talk more about that today. So next slide. Okay, and this presentation was given was inspired by a presentation I gave to the American Association of Petroleum Geologists last year, uh, last fall, as a tribute to my dear friend Luis Pomar, who unfortunately passed away in 2024. But he was an incredible inspiration to so many. Next slide. And some of the personal qualities and abilities that made him such a superb scientist and educator. And these are qualities that will help any geologist to, I encourage you to develop, is the ability to observe and interpret geologic features, a willingness to consider ideas and observations of others, but also to question explanations that don't fit. It if things don't quite fit, uh, and he had, and this is what uh, allowed us to collaborate, is his amazing ability to visualize processes. I can interpret and understand processes. I can interpret them uh, more verbally, but I'm not. Uh, I had two siblings who were artists, and they got all the artist capabilities in my family, so I've very minimal artistic capabilities. So working with Luis was an amazing privilege. Next slide. And there, it's Luis on an outcrop explaining a particular carbonate feature that he probably on the island of Majorca. Next slide. And one of the things that puzzled Luis right up until we published this paper, or until published his last paper, was the rates of carbonates are so much higher during times of high atmospheric carbon dioxide. Much of the limestones that we see have recorded in Earth history were formed during times of high atmospheric carbon dioxide. So he didn't, for many years, didn't get why rising atmospheric carbon dioxide concentrations should cause a decline in carbonate production, particularly a problem for coral reefs. And so he was puzzled by why rising atmospheric carbon dioxide concentrations were a threat to modern coral reefs and other modern carbonate factories. Next slide. But he explored questions visually, and so again, rates of carbonate production and accumulation were typically much higher during times of atmospheric carbon dioxide. And this is one of his amazing interpretive figures. And next slide. Here are just some examples of those limestones that were precipitated during times when carbon dioxide levels were higher, and some actually after, like the early Mississippian mud mounds after May, and the upper Triassic reefs of the Austrian Alps were precipitated during times of elevated carbon dioxide in the atmosphere. Next slide. So, stromatolites are the key to, are a major key to this conundrum, or this paradox. They've occurred through Earth history since something like 3.7 billion years, despite dramatic changes in physical and chemical conditions in aquatic environments. So it's not coral reefs that have occurred through Earth history. It's cyanobacterial and algal accumulations of carbonates. Next slide. So what question that we asked was, what advantages do photosynthesizing aquatic prokaryotes and algae gain by precipitating carbonates? Why, if they're the carbonate producers, what do they gain by precipitating carbonates? Why don't, I mean, many algae, many cyanobacteria, many algae don't do quite well without precipitating carbonates. Next slide. So, an issue that's long sparked my personal interest in carbonates and carbonate production is that linear thinking about carbonates usually results in seemingly unresolvable enigmas or paradoxes. Rather, my experience, and as Janie said, it's a pretty long experience, is that when faced with an apparent paradox, I recommend that you search for the possibility that both sets of observations are have a validity, or the whole range of observations have validity. It's not the observations that don't have validity. It may be the interpretations or the some less obvious factors that you need to recognize, that to find the key to solving whatever mystery that you're pursuing. Next slide. So an example, I was a a young assistant professor at in Texas, uh, and I had the advantage, as was the Permian Basin Society for Economic Paleontologists and Mineralogists, and the local West Texas Geological Society had speakers, brought in professional speakers, um, 3 days, 3 Tuesdays a month. And so I literally got to hear lectures by uh, the who's who of 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 the petroleum industry, but especially with SCPM, the Permian Basin SCPM, the uh, who's who of sedimentologists. And the thing about the Permian Basin of West Texas is that at that time, about a quarter of America's oil and gas came from a 400-kilometer radius or so the of Midland, Texas. Many people have heard of Midland, Texas, and it was the University of Texas Permian Basin was just uh, uh, a short distance away across some bleak desert. So, but anyway, I had the pleasure of meeting Noel James, the famous Noel James, in the Canadian sedimentologist, in 1982, when I got to hear his talk. Uh, and he's right at the beginning, he was talking about carbonates through Earth history, but he started out with a picture of a beautiful coral reef and said, coral reefs occur in warm, clear, nutrient-rich waters. And I had the opportunity to meet and speak with him after his presentation. And I said, wow, that was an amazing presentation, but I, I have a question. How can nutrient-rich surface waters be clear? Wouldn't warm surface waters, if they were if they had if they were nutrient rich, wouldn't there be lots of phytoplankton? And Noel hesitated and then replied, I shouldn't say that, should I? And instead of being offended, here's our AAPG distinguished lecturer acknowledged that my point, and we've had a long-term friendship and collaboration, gone to sea together. Uh, yeah, he's an amazing person. But his recognition uh, and some other issues prompted that paper that Jeannie mentioned, the nutrient excess and the demise of coral reefs and carbonate platforms. And for the young people out there, I teach mostly scientific writing at this point in my career, and academically. And if you have a really important concept, chances are it's going to be harder to publish than it's kind of run of the mill uh, basic research. So my recommendation is don't give up. because that paper, Nutrient Excess and Demise of Coral Reefs and Carbonate Platforms, was rejected outright by both Science and Nature as being of insufficient interest to review. And it was rejected by Coral Reefs as too speculative, wasn't yeah, it's too, it wasn't just a good review paper, it was too speculative. And so fortunately Paleos had just the SAPM had just launched Paleos, and I was able to publish the paper with some really valuable input from Wolfgang Schlager uh, in Paleos in the first volume, third issue, uh, and I have to chuckle every time I think about the fact that it was of insufficient interest uh, to even review in the eyes of the big name researchers. Okay, or big name journals. So next slide. In the mid 1980s, after a presentation on carbonates and larger benthic forams in oligotrophic waters, and the hypothesis of the talk was is that the very complex, very large and specialized benthic forams were specialized to very low nutrient conditions that made for very clear water so that they could essentially divide up the depth habitats down to um, in excess of 100 meters, sometimes in excess of 200 meters in very, very clear water. And an audience member said, I was wrong. That couldn't be true, that there were such nutrient deplete areas of the oceans um, because someone else had just published on the large predators in the eastern tropical Pacific, for example, that occurred at the same time, and they had to have lots of food. So there must not, so I had to be wrong about there being limited food in the areas where carbonates were being produced. And fortunately, that discussion on the on the plane on the way home from the meeting, I basically put together something that I called fluctuations in the trophic resource continuum. Essentially, uh, how essentially it's uh, oceanography. It's paleoceanography, and that is if you slow down ocean circulation, you have a broader range of possible habitats than if you stir the oceans fast. And so uh, and what I perceived from that question or from that uh, criticism was is just thinking about the rates of ocean circulation and how they varied through time, and then how that uh, the upwelling areas were richer, but the oligotrophic areas were poorer, and so the and uh, then during warmer times, greenhouse world times. So next slide. So another uh, carbonate uh, hero of mine, Stan Frost, asked in 1977, why did shallow water coral reefs, which are warm water ecosystems, emerge as dominant carbonate factories in the Cenozoic as the Earth transitioned from the greenhouse world of the Cretaceous and Paleogene to the Neogene icehouse world? So uh, the the Paleocene, Eocene times were not times of prolific coral reefs. In fact, they're the times of really prolific production by algae and larger benthic forams. Next slide. So, and those questions led to, or that, looking at that conundrum or that paradox and exploring it with Luis Pomar and his incredible ability to visualize phenomenon, we published a range of papers, including specifically on the paleoceanographic evidence for changing habitats for larger benthic forams. Next slide. So Norman Newell, another of the historic the heroes of paleoceanography or paleontology. Mass extinction events in the fossil record are characterized by global carbonate depositional hiatuses. He made that observation and numerous times, but in print for sure in 1967. And so the major mass extinction events are times of rapid increases in carbon dioxide in the atmosphere and other acidic components that makes the systems inhospitable for major carbonate depositional systems. But if you look at the details at the extinction horizons, and you look before the extinction event, for example, the fossiliferous limestones of the late Permian carbonate strata, that what overlies them are microbialites and oolites. And in the Cretaceous extinctions, post-Cretaceous extinctions, you have echinoid gastropod packstone and dolostones with calcareous algae, basically Dasyclea and Codiaceans. And again, that you shift, the shift is often from mixed plant animal, I guess is the best easy way to think of it, systems to dominant autotrophic and heterotrophic. So from mixotrophic systems tend to be extremely more fossiliferous limestones with more metazoan type fossils to your more but metazoans that were probably algal symbiont bearing to your pure autotrophic heterotrophic carbonates. Next slide. Hopefully I'll make that a little clearer. And so the final paper that Luis Pomar was able to publish, and before he was declining from Parkinson's disease, and Guillaume Matteo Vicens, his highly successful PhD student whom I had the privilege of being on GAM's graduate committee, and GAM spent some extended periods of time working with me and as during his PhD work, and also his close friend Juan Buceta, Juan Ignacio Buceta. They helped him. We all helped him get this final paper published. And the first part, and this is Luis's Luis's synthesis, that he proposed the phosphate extraction mechanism, and and it explains to explain why calcifying microbial and algal biota thrive in oligotrophic conditions. Next slide. And the answer, or the light bulb, the insight, is phosphate. Phosphate is bioavailable phosphate is essential for all life forms, and phosphate deficient environments tend to be the very clear water, uh, low primary productivity areas in the oceans. And but phosphate was much more abundant earlier in Earth history, and such that in the Archean it was uh, well, as cellular structures were evolving and developing, it's phosphate is essential to nucleotides, phospholipid membranes, energy transfer and storage, and synthesis of proteins and enzymes. And in other words, phosphate is essential to life processes. But it has received less attention in research than carbon, nitrogen, and iron. And of course, everybody thinks about iron right now, and nitrogen, but it's especially uh, in the evolution of autotrophy in the Neoproterozoic. It was the decline of phosphate, and basically as those early uh, as those early photosynthesizers started poisoning the environment by producing all that oxygen, phosphate became much more limited because of its low solubility in oxygenated waters. And there's the conundrum, or the paradox, to something that to think about on the side, and that is that oxygen, the cellular structures were developed in low oxygen times in Earth history, or in the very low oxygen times in early Earth history. And oxygen is actually especially oxygen radicals are actually toxic to cellular structures, which is what goes on with coral bleaching, in fact. So anyway, phosphate has low solubility in oxygenated waters, and therefore is an essential limiting, perhaps, in many cases, essential limiting nutrient for primary productivity in Phanerozoic marine ecosystems. A lot of people like to talk about iron being the limitation, but phosphates right in there, and cyanobacteria can blue-green algae or cyanobacteria can fix nitrogen from atmospheric nitrogen, or N2, uh, but uh, but they phosphate is quite limiting, as is iron. Next slide. And so again, this is one of Luis's visuals, and showing the change in phosphorites formation between the Ediacaran and the Cambrian, when phosphorus, the part per million of phosphates, dropped dramatically with the uh, end of the Neoproterozoic and the early Cambrian. And next slide. And what I mean, Luis did not invent this, or didn't this phosphate extraction mechanism. He just realized how important, and how this was the key to carbonates. And that is, especially algal carbonates, is that the co-precipitation of phosphate and calcium carbonate is that calcium carbonate can be both a sink and a source of phosphate for photosynthesis. And that is, and so in a very phosphorus, low nutrient limited, or nutrient limited, or phosphate limited conditions, the precipitation of calcium carbonate in daylight actually sequesters a certain amount of phosphate precipitation in a carbonate structure, in the carbonate crystal structure. But then at night, under the lower oxygen conditions, especially in the uh, structure of the biocarbonate, the phosphate uh, can much of that phosphate can be desorbed, and therefore taken up by the algae to be used in photosynthesis the next day. And so uh, the essentially the phosphate, some of the phosphate that's adsorbed, I shouldn't say absorbed, adsorbed onto the precipitated carbonate during one day, then is much of that phosphate is desorbed and made available for the cellular structure to utilize that, to retain it and then utilize it in photosynthesis the following day. Next slide. So the second part of that, the adsorption desorption process was what Luis read about. But then the next question was, how did that, what did that do? What's the connection with biocarbonates? And so this was an area that Guillaume and I helped him especially with in this final paper. And rapid increases in acidity, whether it's of a volcanic or a meteor impact or human effects, rapid increases in acidity result in hiatuses in carbonate production, which addresses Norman Newell's 1967 observation. But it also, because carbonate production is controlled by a range of physical, chemical, and biological processes, that addresses Stan Frost's question of, why So next slide. Okay, a key factor in this story is calcium carbonate mineralogy, and that is that aragonite is stronger than calcite, but it's less stable and more soluble, and so in the fossil record, it's more poorly preserved, but also from a from a hydrocarbon exploration standpoint, it promotes the dissolution of the aragonite, or the recrystallization of the aragonite, uh, uh, promotes porosity, whereas calcite precipitation is inhibited by uh, magnesium iron by magnesium ions that essentially interfere to a certain extent with the precipitation of calcite, of essentially calcium carbonate. But once it's precipitated as calcite, once calcite is precipitated as at least as low magnesium calcite, it's relatively stable. And so, our fossil record for the most part is calcite. And but the calcite uh, is results are the uh, the production of calcite and then the fossilization results in minimum porosity, whereas mag calcite, essentially variable magnesium calcite, and so depending on how much magnesium is around, the there are certain groups of organisms that can just go ahead and especially many algae, but many forams and other things can actually precipitate magnesium, not generally fully in equilibrium with what there's some exclusion of magnesium ions, but uh, they they're not excluding as much of the magnesium. And so actually the magnesium calcite uh, can vary in the amount of magnesium depending on the amount of the calcium magnesium ratios in the seawater. And of course, high mag calcite often dolomitizes, which can promote porosity. And so that's one of the reasons of the carbonate mineralogy is of interest to the oil industry, which is where a lot of the carbonate studies have been done over history. Next slide. Okay, and so just some of our modern aragonite producers. There are calcareous green algae, Halimeda, and other calcareous greens, and of course scleractinian corals and some others. But especially those are the two big aragonite producers in modern oceans. Next slide. The calcite producers with that actually actively exclude the magnesium in the precipitation of calcium carbonate include coccolithophorids, planktic forams, and many, but not all, benthic forams. Not even all the rhodolines. Next slide. There's a whole bunch of things that can do variable magnesium calcite precipitation, such as octocorals and uh, miliolid forams and calcarinid forams, and crinoids and echinoids and some kinds of not and your actual coral and algae, the coral and algae as opposed to calcareous greens. So the coral and reds as opposed to calcareous greens. And so um, they are not as limited by the amount of magnesium in the water. Next slide. Which brings to the important paper of Sandberg and then Stanley and Hardy, and Sandberg 83 and Stanley and Hardy in 99, who recognized that the importance of the aragonite, the calcite aragonite cycles in Earth history, and uh, and that aragonite and high mag calcite times were quite different from uh, the calcite uh, uh, low mag uh, calcite uh, times in Earth history. So next slide. So in the Phanerozoic, our greenhouse conditions generally involved extended times of relatively high CO2, Silurian, Devonian, Cretaceous. They're characterized by thick and extensive accumulations of fine-grained carbonates, and the dominant precipitation was cyanobacterial and algal calcification, but often baffled by skeletal carbonates, by not in addition to, well, commonly baffled by uh, skeletal carbonates produced by, in many cases, metazoans, but in some cases also larger benthic forams. Next slide. Icehouse conditions, at least our modern icehouse conditions, in the Cenozoic, they began developing in the Oligocene, and this is in paleoceanography. We know that the Oligocene was a time of, of kind of dramatic change in the Earth systems, and but interestingly enough, coral reefs emerged as major carbonate factories uh, under icehouse conditions. And this is one of those conundrums. So climatic and geochemical influences on biological processes is that slightly cooler climates favored algal symbiosis. One of the things we're seeing right now is that um, coral bleaching, for example, or foram bleaching in the larger benthic forams, the symbiont bearing larger benthic forams, uh, is actually a photooxidative stress response. It's the at warmer temperatures, more warmer temperatures or shorter wavelengths of solar radiation, the more oxygen radicals are produced, which then cause damage to the cellular structures of larger benthic forams, and actually the cellular structures and of uh, the corals themselves, and um, and as the magnesium calcium ratios increased in seawater, that actually favored production of aragonite and aragonite hypercalcification by corals and calcareous algae. But coral and algae that produce magnesium calcite also thrived. And so under icehouse conditions, but in the warmer areas of the oceans under icehouse conditions, conditions optimize calcification and phosphate extraction. Next slide. So that kind of resolves, or that I encourage you to read the Pomar et al. 2022 paper, because I probably haven't done an adequate job here, but microbial and algal produced carbonates have persisted through the geologic record. Yes, they've been knocked out, or they lose dominance to non-carbonate producing microbial and algal conditions during times and places of higher nutrients, higher especially higher nutrients, and so that phosphate extraction isn't an important factor. But then but it also helps us understand, if understanding phosphates helps us understand why microbial and algal produced carbonates are there everywhere we look since the Proterozoic, whereas under rising atmospheric CO2 conditions in the modern world, that's a threat to our modern carbonate factories, which have evolved to as this symbiotic relationship between, instead of the calcification being done by the algae themselves, the calcification is being done by the coral structure, but that calcification is sequestering a certain amount of phosphate that then can be released to support the algal symbiosis. And one of the pieces which in my early research was showing that if a coral or a larger foram has plenty of sunlight, but not so much that it's damaging, if they've got sufficient sunlight, they can produce almost unlimited amounts of organic matter, of simple sugars, junk food. And it's been called junk food in the research, in the publication record, because there's plenty of energy. The algae, the symbiotic algae, can produce lots and lots of simple sugars for the energy for the corals, and the energy to precipitate more calcium carbonate, but they provide the energy to precipitate the calcium carbonate that then incorporates a certain amount of phosphate that then allows the growth of the system. So next slide. So that's pretty much what I have to say. I hope that was useful and made some sense, and please excuse me if it doesn't, because it's 2 o'clock in the morning my time.
Okay. I think we can open up. Aditi, are you there?
Yes, I'm here. I think you can. I've stopped sharing the screen. I hope it's still not bothering anyone.
No, I think it was a very nice explanation where she tried to explain that why, when carbon, the main thing is whenever there have been, we are all now saying that because of carbon dioxide increase, there is more acidification and there's more reduction in the, a threat to our coral reefs and the carbon dioxide or the biocalcifiers. whereas her study very clearly shows that actually, if we look throughout the geological record, whenever there has been more carbon dioxide, you are actually finding more carbonate deposits.
Yeah.
But there is a difference in the high Mg calcite and the low Mg calcite, which has varied with ice house when carbon dioxide emissions are low, and vice versa. So I think uh, we could probably open this up for if there are any questions, Aditi, that you already have, or we should invite people to ask
Yeah,
questions.
I think people can just unmute themselves and ask. I have a question I would like to ask actually.
Yes.
Okay.
So,
Yes, Aditi.
what I was interested in is your analysis of how phosphate limitation also has an impact on biogeochemistry of a particular region. And my question is, in the ocean warming scenario where we are already seeing effects of uh, well, ocean acidification, I'll speak as a layperson, but how do these phosphate chemistries and iron limitations would change in the face of ocean warming is what I would like to know. I mean, do we have some understanding of what would happen?
Well, we know that um, it's we know that the uh, in the short run, or the short run geologically, it's a short time frame geologically. That in fact, that's something I should have probably included in the talk, is the discussion about the length of time of the mass extinction events, and the length of time that uh, that it was sufficient acidification to suppress carbonate production essentially globally for some extended period of time, uh, thousands of years, or tens of thousands of years, versus millions of years with the uh, Permian uh, extinctions. And of course, uh, uh, yeah, the am I answering this question right, or is my is my uh yeah. And so the length of time that it takes. The thing is that um in the long run, I guess, once the carbonate factory, the algal carbonate factory kicks in fully, but or even before even before it kicks in with carbonate, it's the photosynthesis, the very process of photosynthesis, um, removes uh, CO2 from the oceans and atmosphere. And so in the more oligotrophic parts of the ocean, you have not only um, the photosynthesis, but the photosynthesis that is resulting in the production of microbial and algal calcium carbonate, which is another mechanism for removing CO2 from the system. Yes, you know, there's that conundrum, that paradox, that for every calcium carbonate that gets precipitated, uh, 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 there's a CO2 that gets released when you think of bicarbonates and to carbonates. But but that's a whole uh, another discussion about yeah, short term versus longer term effects of excess CO2. I'm not sure I answered your question, but that's yes, it
I
is.
mean, sort of. I'm new to the subject. I'm new to learning biogeochemistry, but what I'm understanding is basically that the carbonate chemistry, the phosphate chemistry, they actually work on different time scales, and as a result, the manner in which they interact as a system in any system are will not very well understood, and that is the reason. I mean, I don't know if I'm getting it correct or not.
Okay, no, the abundant phosphate, the amount of phosphate in the systems, in the early Earth systems, was much higher. And but as cyanobacteria, as algae, cyanobacteria, and ultimately microalgae evolved, they caused the first great pollution event, if you want to think of it, because they produced all this oxygen that all these anaerobic organisms couldn't handle, and and so they had to go underground or into high nutrient areas where they could where there was or higher nutrient, higher organic carbon, lower oxygen conditions. So
Phosphate
I,
was precipitated out and removed from circulation, and
well,
now, yeah, so it's a sediment. It can only come back into the system through sedimentary cycles. So in the
well,
early waters, there was more phosphates than now.
yeah, a major source of phosphate is volcanism.
Igneous rocks or
Yeah,
recycled sedimentary rocks,
yeah,
either of the two. Yeah.
Yeah, that's a bit clearer. Thank you. I
Yeah.
need to
So,
do my end of reading as well.
Rama,
Uh,
yes. Any
I
more
think
questions?
yeah, Supriya sir had unmuted himself, but I'm sorry about
Yes.
that.
Yeah, just very nice talk that you have explained how the paradox was solved. But this another question, relevant question, comes into the current context. For example, we know that during the Cretaceous, carbon dioxide was very high compared to today, and carbonate precipitation was also very high. That in fact actually controlled the Earth's climate, acting as a thermostat. So in current condition, we already know that increased amount of CO2 is increasing acidity and also acid affecting the coral's growth and coral's ecosystem. So can we have this kind of conundrum again and solve so that you can reverse the effect, global, the rising temperature effect on coral reef ecosystem and as an in turn on Earth's climate?
okay. I'm not sure I understand or that I got all of the question, but the one issue is the greenhouse conditions, the rising temperatures are a key piece of that well, actually, that's let me drop back. Remember, I said that oxygen is toxic. Oxygen is toxic under certain
Methanogens.
conditions, or under at the and the thing is that our cells, at least as I understand it, our cells are uh an archean, not archean, and a yeah, archaea, not archean,
Archaea.
archaea.
Methanogenic bacteria. Yeah,
Yeah,
methanogenic bacteria.
yeah,
Archaea.
yeah. And the with the mitochondria, which is the symbiosis and cell evolution concept, which is finally really broke through in the 80s, I guess, and understanding, but it helps us understand why oxygen is toxic to the cell when too much oxygen radicals, it's not the oxygen itself so much as the oxygen radicals, because and here we get into a whole issue of the cellular system of photosynthesis, and in photosynthesis, if there's too much my brain train of thought just jumped this track, but the our cells are quite primitive in the sense that oxygen, there's not a good, it's Rubisco, that's what I'm trying to come up with. Rubisco is not doesn't distinguish well between oxygen and CO2. And so when it at warmer temperatures and higher light levels, the Rubisco, actually what gets produced are too many oxygen radicals. It increases the
Okay.
production of oxygen radicals, which is then causes the intracellular damage. And in the
Oh,
forearms, it just causes degradation within that part of the cell. Whereas in corals, there's some degradation, intracellular degradation, but the main thing is that it
oh, okay,
somehow
okay.
the triggers the coral to expel the algae,
The algae, the algae.
to reduce the rate of photosynthesis, or the rate of production of oxygen radicals. That's what bleaching is to get rid of the get rid of the
The
oxygen
algae.
radical producers, basically. And
Okay.
so now there are corals, for example, and their forams for that matter, the calcarinid forams, that are quite tolerant of higher temperatures, but generally speaking, they are not the uh, high carbonate producers. They have the slower growth rates, and for example, if you go to the Florida Reef Tract, which was once, I don't know, 20-30% coral cover, now it's less than 10%. And the corals that are remaining are not the the historic reef producers, the acroporas uh, and so forth, and so um,
Okay.
this is
So
this
Pam
is
here,
basic ecology. It's ecology in that you've got to have the chemistry, the physics, the biology, and all of this is controlled through geologic time, through evolution. So
okay,
it's all ecology. I'm sorry. It's all.
okay. So one something similar. You were just saying that whenever there is increased temperature, there is more production of oxygen radicals within the cellular body, because of which so it's not just the carbon dioxide, but it's also because of the temperature, the radical oxygen radical production, which leads to expelling of the algae, causing death of the coral reefs or degradation of the coral reefs. So it has
Right,
got something to do with the temperature, which is a byproduct of the increase in carbon dioxide.
right. Yeah.
But then, But then I also want to know, right, what we are talking about is increase in carbon dioxide levels are bringing down the pH of the waters,
Yes.
right? But then do coral reefs and maybe larger foraminifera, because they host the symbionts which use up carbon dioxide, okay, does it act as a buffer because they're using carbon dioxide for photosynthesis? So shouldn't they
Mm.
actually lead to more acidification?
Okay.
Because
The okay, the modern, okay, there are the corals that evolved in the coral lineages that evolved in the Mesozoic. There are
mm,
coral lineages, but many of those that are still extant are the ones that are slower growing or deeper water or whatever.
mm, mm,
They're not the prolific reef builders. And
okay,
so it's partly that the 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 really prolific reef building corals that are extant today, but are the ones that are in trouble today, that are being they are the ones that they are more sensitive to the lower pH.
okay,
The thing is that, and different organisms, but if you think about it, like the corals, the more ancient corals that might have evolved during higher CO2 levels, the the larger forams that evolved and were prolific carbonate producers in the uh, uh, Paleogene, those the other thing is that um, it's kind of interesting that experiments, especially with the larger forams, people who've done experiments trying to see uh, their
yeah,
the research was all over the place in the uh, aughts and teens of when people would do ocean acidification experiments with forams, with symbiote bearing forams, and they were finding it that if they really at 2500 ppm or something like that, they could suppress the calcification of the forams, but but up to about 1,000 or even 1,200, they
carbonate
didn't suppress
production was
the
not
forams.
suppressed.
The forams did fine at intermediate increases. And so this is probably an evolutionary uh, difference between, and then just like, again, some of the corals, uh, the more ancient corals uh,
Were more resistant
can
than
tolerate,
those today.
yeah,
Yeah,
it's the ones
they were
that
more
it's the
tolerant than
ones,
those today.
yeah,
Okay,
the ones that evolved in the Neogene. Those are the ones, the really prolific reef builders of the Neogene are the ones that are the most sensitive to the
more
increasing
threatened. Yes.
CO2
Okay.
and to the increased photooxidative stress, so forth.
Photo-oxidative stress is what. Yes. Any more questions, Katal sir, Nigam sir?
I see Omkar has his hand raised, and I also saw Mugdha raising her hand. So maybe we can have Mugdha first and then Omkar, if that's okay.
Hi,
Mugdha?
ma'am. Yes. Actually, I got my answer. Supriyo sir already asked my question. So, uh, I saw the paper by Pomer et al. 2022, and where he has explained how carbonate precipitation was caused during the high CO2 conditions. but the study is based on the Archean sediments, and the oceanic conditions were different then than they are now. So, can that logic be applied in the current situation?
Okay, I missed the first part. What was the
Um, she says that the Pomar et al. Paper talks about conditions in the Archean,
right?
Yeah.
right? So during the Archean, the ocean chemistry and the ocean setup was so different.
Oh, yeah, and that's
So,
the whole
so,
point. That's
so
the whole point, is our cellular structure evolved in the Archean. Cellular structures, and the especially Rubisco is very conservative, and it's there's quite, I mean, I'm not a good enough botanist to understand the whole reasons for why Rubisco
more sensitive
as
now.
is more sensitive. Well, it's what it is that it's um the ocean chemistry was just completely different when cellular structures evolved.
So can we
And
use the same mechanism
Hmm.
of calcification and dissolution in modern oceans? That is what the question is.
we're not trying to use, well, you can use the same argument for calcification by cyanobacteria and algae, uh, but there weren't the metazoans then to do the calcification.
Okay.
Okay, fine.
So, the like the current situation where, like, I'm studying ocean acidification, and I'm looking at pteropods which are arachnitic in nature, and they are said to be more prone to acidification in the modern sea because they are arachnitic, and they are more unstable because they are made up of aragonite. But like, we have gone through the literature and we have seen that pteropods are more resilient to ocean acidification, and then there are various processes that occur in the ocean which actually uh, it stabilizes the environment. For example, pteropods, they dissolve, and the carbonates are dissolved into the water, and it actually increases the pH of the deeper waters, which actually makes forams more uh, which actually helps forams get preserved in the deeper water because the pteropods, they dissolve
Dissolve in the shallow water. Yeah.
shallow water. So even in the past, when the CO2 levels were high, and it caused carbonate precipitation, but that is not happening in the current situation, and the mechanism has evolved with the time. So we can't really say that oceans are not prone to ocean acidification now, because the mechanisms have changed over time.
Actually, you've got to throw in a whole nother piece of the story, and that is alkalinity.
Yes.
Alkalinity, yes.
Yeah, because uh, for example, um, um, with some Mediterranean scholars, we published a paper a couple of years ago now, I guess, explaining uh, why the why the forams are making beaches in the eastern Mediterranean, uh, in uh, in areas that you would think that I mean, at a time when CO2 levels are going up, but if your natural alkalinity is much higher, if the alkalinity is much higher, the CO2 is coming coming into the environment. The CO2 is a major piece
Yes.
of of alkalinity, and so that's one of the paradoxes, the conundrums of what we're talking about is that the
Dissolution
the
also leads to
actual
increase in alkalinity.
There
yeah,
are so many
it's
aspects and factors which are involved in ocean acidification. It's a whole conundrum, and there's so much to be done.
well,
But
there's just another thing that this made me think of. Your question made me think about, in the thinking about
uh,
the what's going on today in the Eastern Mediterranean. You've got two other factors in the Eastern Mediterranean. What's the geology in the rocks in the Eastern Mediterranean? Volcanics and limestones, or
limestone.
limestones,
Lots of
dolomites.
calcium and phosphates coming out.
Yeah, so well, it's not even so much the phosphate, it's they're providing the calcium, the dissolved calcium, to go with the alkalinity and then the forams, which are adapted to uh thousand
Yes.
parts per
I
million.
think that is the problem. We are looking at Arabian Sea where the processes are much different, and in Mediterranean it
Exactly.
is much different. So we cannot just have a uniform processes or effects all over the
No,
place.
Okay.
you don't. And
Any
yeah.
more? Who goes next? Any more questions?
There was a gentleman had a question.
Yeah.
Kathal sir wants to ask something. Kathal sir wants to ask something.
Uh-huh.
So
Yeah,
please.
yeah. Professor Palm, ever since I read your paper on the symbiote bearing foraminifera, since then I had a question in my mind and I get an opportunity to ask you today. Could you date the start of symbiote bearing process? When it started?
When did the symbiont, I'm sorry,
Symbiote bearing. When the symbiote bearing habitat started in foraminifera? I mean, do we have any data? I mean,
most,
when could it start?
Haven't
well,
haven't they always been there? So whenever you've had these larger foraminifera or those associated with coral reefs, I think ever since that time we've had cement bearing foraminifera.
Do
Permopan,
we have any date? In Miocene or in Eocene, in which period they started?
Even
Is there
before.
any
well,
data?
no,
I'm simply curious to know.
no, they were there were symbiont bearing forams. Certainly the fusulinids probably had algal symbionts in the permopan, but then in the I'm I'm pretty ignorant of the foram assemblages in the Cretaceous, okay, or
Okay.
in the Mesozoic, but the larger benthic forams evolved like crazy in the Paleocene, Paleogene, and so especially the Paleocene Eocene times. I mean, they uh, that was a time when they were
Probably had
and that's
started.
why and that's when things that are close to some of our modern forms, but again, they lived at a time perhaps when the alkalinity overall was much higher, and so they could tolerate the higher CO2 levels as well,
Okay.
and just like the Mediterranean today, or even probably the Gulf of Aqaba and some of the Middle Eastern high salinity areas, essentially the elevated salinity up to a point is advantageous in terms of alkalinity.
Okay, fine.
And
Thank
I
you.
think I think Permian, I think Carboniferous Permian were the time when 350 million years. I think so, sir.
yeah, but there have been,
Yes.
I mean, symbiosis, symbiosis is why,
Good.
is why anything
Yes.
is here except for
Yeah.
Yeah, that is
cyanobacteria
good.
and archaea.
Any more questions, Aditi?
I think Omkar and there was another student, Nishant, who had raised their hands. So these will be the last two
Nishant? Yes,
questions
I think
we take.
Nishant
Yeah.
wants to ask a question.
Mhm.
Yeah, actually, I have two questions. My first question would be like out of curiosity about. So one of the initial paradoxes you while you were answering, there was one point about turbulent events over sedimentary records. I would just like to know how much of like these turbulent events actually show up in sedimentary records, and if they show up significantly, how much of exactly is being contributed because of internal waves, if they contribute significantly.
Okay. The we on the West Florida shelf in the West, just an example, but um, we have some deep reefs, so-called deep reefs. They're deep, they're reef structures that are at the lower end of the photic zone. And and actually if you think about even the Cretaceous, the rudis were the shallow water carbonate producers or Metazoan carbonate producers. The corals tended to be in deeper environments. And and if you think you're asking, and to make sure that I understand your question, you're asking about internal waves and how prevalent they are in it was that what was your question about internal waves?
Yes, yes.
Okay, yeah. I think internal waves are incredibly important relative to the so-called um uh mesophotic reefs, or the sort of the deep euphotic zone,
Yeah.
because the internal waves are bringing up food, but
Yeah.
they're also they're
Agitating the ocean.
also when you reduce the pressure, the carbonate saturation increases
Mhm.
as the wave breaks over the deep shelf.
Topography. Yeah.
Does that make sense?
Yeah,
Yeah.
yeah, yeah, yeah,
Yeah,
definitely.
so and it's something, I mean, we had some of the people in my college have been very active in developing gliders and work on the West Florida shelf, and some of my retired colleagues work very much on the deep reefs on the West Florida shelf. And I've always thought that that was a perfect that was a research area just waiting for somebody to analyze both the oceanographic data to go with the uh, biological data. That, for example, there's an area on the West Florida shelf where the deep reef, it's as it extends north, it grades into a octocoral habitat
Yeah.
from a carbonate, essentially from getting your light from above to getting your food from catching the
From
Yeah.
food
below.
in,
Hmm.
and from with and the internal waves certainly are also carrying more plankton, and
Yes.
so there's this an interesting there are projects to me, there's chemistry projects just waiting to be explored in this gradient between the deep coral reefs and then where they give way to the octocoral reefs or octocoral habitat.
Yeah.
And of course, you've got the third factor, and that is that the carbonate habitat, the carbonate habitat was in the reduced at lower sea level stands.
Right. Yeah.
Yes.
Yeah. And another question would be, I would just
Nishant,
like to, yeah, sorry.
Nishant, I'm sorry. We've had Omkar waiting for quite a while. So
Oh,
can he
sure,
ask the
sure, sure.
question first and
Yeah,
we can come
yeah,
back to you if there's more
sure, sure,
time?
sure, sure. Yeah,
Omkar,
sure.
are you there?
Yeah, I'm there.
Yeah,
I just had
sure.
a simple question. Like now we have seen a large amount of what plastic pollution is increasing, because I just wanted your views on plastic pollution, because these are directly affecting the plankton foramens on the sinking rates. And so what are your views, like how it's, at what scale is the plastic pollution affecting the plankton forams and the other processes as well?
Well, you're way beyond my expertise on this because my expertise with plastic pollution is picking up trash when I walk my dogs. and or I have a net or I have a long pole that I retrieved in a coastal cleanup one time that I've retrieved balloons or plastic bags or whatever that I live on Tampa Bay and I can but my I am horrified by the what plastic pollution does, and so I try to fight it at every level, but I have no clue.
No
I
idea
have
how it
no
affects
experience,
settling
no
plankton
ideas
foramen.
about
Yes, agree.
what it's doing with plankton forams.
Yes,
Probably
agree.
not good.
Okay, yeah,
Okay,
fine.
fine.
Maybe you can come back to Nishant if
Yeah,
anyone else
Nishant,
doesn't have a question.
Okay.
you had another
Yeah, I
question,
just,
Nishant?
I
I would
think we'll
just
end with
like
this
to,
question. Yeah, last question by Nishant.
okay,
Yes.
I would just like to know about your take on this geoengineering problem of trying to tackle ocean acidification using by increasing ocean alkalinity. There have been some efforts going on,
We've done that.
which I'm exposed to recently. So I
Pam,
was
we've
just
been
curious
speaking
about your
about
take on
it,
it.
right? We started with
Sorry?
our project with, this was a beginning to one of the Fulbright project that we started, where people are
Uh,
offering solutions of bringing in a lot of bicarbonate or bringing in a lot of calcium carbonate and dumping it in the ocean, by which you
yeah,
can probably tackle the ocean acidification problem. What do you have to say about
yes,
it?
yeah. Some of my own professors are working on this problem. So I was just curious about your take on it.
Well, hmm. uh, I guess increased heat increases the evaporation rates, which increases the salinity of the surface waters, which increases the alkalinity, and also increases the uh, erosion rate. So the dissolution of it in over land. So it increases uh, the uh, increases the ions in your the dissolved matter in your freshwater systems as well, and so and increasing the alkalinity. If you think about it, it's increasing the alkalinity that that ultimately helps solves the solves the problem of the ocean acidification and allowing the cyanobacteria and or the the cyanobacteria and the planktic algae and the microalgae and so forth to actually precipitate the carbonate and ultimately
Nishant, the
the green
complete,
algae.
yeah,
Yeah,
in this, it completely depends, which he always has said. It depends on which direction the equation is going. Are we
yeah,
Yeah.
splitting? Because with every calcification, with every calcium carbonate being formed, one H plus ion is being released. Whenever
Yes.
you break up the bicarbonate, okay, you are releasing an H plus ion. So actually
Yes.
thinking that calcification leads to more acidity. Okay, so it
in the
Okay.
depends
short run.
on, so it depends on which side of the equation is going faster, and that
Right.
depends on fresh water. Is there more fresh water influx, or is there more evaporation? What,
Right.
at which end of the equation? And this varies geographically from place to place.
Right. Right.
So
Yeah.
if you're doing it at one place, it is surely affecting adversely in some other place, somewhere very close by. So that is
Right.
something that whenever we are doing any solutions like this, it needs to be looked at.
Right. Right. Yeah. Yeah. Thank you.
Mhm. Thank you for
Yeah.
the question.
Aditi,
All right.
I think
Yes.
we can, it's
Yeah, we can
3:20.
close. I hope everyone has had a good talk. So I'll be
Vote of
proposing
thanks.
the vote of thanks. First and foremost, I want to thank Dr. Pamela Hallock Muller for agreeing to give an incredible and inspiring talk for our World Oceans Day webinar. Uh, it's always a source of great inspiration for me to see women not just doing well in science but in leadership roles, and just having women speak about their experience and their work is extremely inspiring. I know from my personal experience. So I really, it means a lot to me that you agreed to speak today. Thank you so much for that. I also want to thank the Ocean Society of India, GC, and Dr. Kurian for always supporting our endeavors and our activities. Many thanks also to Dr. Supriyo for his encouragement and always being there. I literally asked him today morning if he could speak, and he just said yes immediately. Um, Big thank you also to Dr. Rajni for introducing Dr. Pamela to us and always being ready to participate and help out with all our activities. And also, I want to thank all the office bearers and members of OSI Pune chapter for their support. They're always encouraging any activity that is suggested and always helping us improve on our activities. Last but not the least, I want to thank all those who are present today. We've had so many stalwarts attending the talk as well as students, and it's always important that some students take away some message or some inspiration from each of our talks. So I apologize if I've missed out on thanking anyone, but thank you all for being there. I want to end by encouraging everyone to please join the Ocean Society of India as life members. On our website, the forms are all available. It's a wonderful community who are all extremely passionate about our oceans, about conserving them, about protecting them, and just being a part of that community is really, really wonderful. So please do consider becoming a life member. You can go to our website, Ocean Society of India, and there are, of course, all the details of joining, and all our activities are also updated over there. So thank you so much, everyone, and thank you so much, Dr. Pamela, for staying awake till the middle of the night, so
I
late that
hope
it's
you have a
almost.
longer sleep tomorrow morning. I,
Yeah.
I hope you sleep longer.
Yeah, well, my husband has already promised to walk the dogs.
Oh,
That's
nice.
great. Yeah. All
Thank
right.
you so
So
much to
I'm going to
join.
stop recording now so that,
Yeah.
yes.